Dr. Rayid Abdulqawi. He is a consultant in transplant pulmonology, consultant transplant pulmonologist at King Faisal Specialist Hospital and Research Center in Riyadh, and he is the adjunct associate professor, College of Medicine at the Faisal University, who will be talking to us on the genetic basis of idiopathic pulmonary fibrosis. Dr. Rayid, please go ahead.
So I thought to give myself a presentation related to genetics because I think, I don't know whether it's going to be at our time or in the generation after, that the way that we practice medicine nowadays is not going to be relevant in the future. So we are moving towards artificial intelligence and genomics affecting all aspects of medicine. So actually, when we think about idiopathic pulmonary fibrosis, back in the 1950, there was a report at the Chest Journal where the title was IPF is occurrence in identical twin sisters. So this was the first evidence that IPF has a link to genetics. And in 2000, there was a start of a very ambitious program in the US, and it was an international effort, the Human Genome Project. And the aim was to sequence the human genome, and it went through many phases. So the first 5 years was to develop the technology for sequencing the genome and build the computational power for the genomes. And in 1997, they recruited the volunteers to sequence their genome. So some facts that I wasn't aware until recently, that if you are to say the 4 chemical bases of the genomes, the A, T, C, and G, it would take you 100 years to finish reading the chemical bases. So, subhanAllah, we have very complex genome. And it wasn't really possible to sequence the human genome without the international effort for that. So, and in 2001, the first draft of the human genome was published. And in 2003, the final report of the human genome was published. And then was followed by the 1000 Genome Project that was announced in Cambridge, UK, because you remember the Human Genome Project was just based on a small number of subjects. And the ambition was to have more subjects to get more insight into the genome of the human, and they ended up with over 2,000 subjects sequenced. And in 2009, the 3 scientists that you see the pictures here, they were awarded the Nobel Prize for the discovery of how chromosomes are protected by telomeres and the enzyme telomerase. So telomeres are the structures at the end of the chromosomes, just like the cap on your shoelaces, because with each cell division, the body is not able to replicate the whole of the DNA. So with each division, the telomeres get shorter and shorter, and until if you don't do anything about it, then basically then the cell will stop dividing, because there will be degradation of the DNA, and then that will lead to the aging and cell senescence and apoptosis. But subhanAllah, our bodies are equipped with the enzyme telomerase, where the body is replenishing the telomere sequences. There are 6 tandem sequences, A, T, G, G, G. So that will enable the cells to continue dividing. And as you can see in this figure, that the telomeres are these cap structures at the end of the chromosomes. And when you have shortened telomeres beyond the critical point, then that will lead to aging of organs manifesting as pulmonary fibrosis, liver cirrhosis, bone marrow failure, and even cancers. And if you look at IPF patients, you see that here that they have short telomeres in around 30% of the subjects. So not even just those with mutations related to the telomere genes, even the sporadic cases of IPF, around 30% of the cases are with short telomeres. And why is that important? Because one is that once you have a short telomere, then you are at risk of rapid progression of the disease. And it's well established that those IPF or lung fibrosis patients with short telomeres, they are at risk of rapid deterioration and decreased time to death or transplant. And we all remember the Panther IPF study where we were until that point practicing giving the triple medication, prednisone, azathioprine, and N-acetylcysteine. And then until 2012, when the result of the Panther IPF study was published, showing that we were doing harm to those patients. But actually, when you look at those subjects where they had DNA on them, and if you look at the telomere length on those subjects, it's only those with the telomere length below the 10th percentile age adjusted, they experienced the harmful effect of the immune suppression. So on the left side, you can see the red line is the placebo, and the blue dotted line is the immune suppressive treatment. And you can see how harmful the immune suppression was in terms of decreasing the survival. However, for those subjects with telomere length above the 10th percentile of the general population, there was no harmful effect. So the message here is that the short telomeres and immunosuppressions are harmful. So there's an interaction between the two. And this was replicated as well in other lung fibrosis cases. So if you look here, even the fibrotic hypersensitivity pneumonitis or the unclassified interstitial lung disease, those subjects with a short telomere below the 10th percentile, they are at a six-fold increased risk of harm, death. Whereas those with telomere length above the 10th percentile, then at least there was no harm or there's no benefit. But the message is here that if you have a short telomere, then you're actually harming patients by giving them immune suppression. And those patients below the 10th percentile telomere length, they experience very rapid decline in their lung fibrosis, as you can see here, even without immune suppression on the right-hand side of the table. So if you had less than 10th percentile subjects, the decline per year in cases of fibrotic hypersensitivity pneumonitis was 111 mils, whereas those with normal telomere length, the decline was only 76. So those with short telomere fibrotic lung disease, they rapidly progress, and we harm them by administering immune suppressive medications. And this has led to a recommendation that we should practice precision medicine. So at least for fibrotic hypersensitivity pneumonitis, when you know that the telomere length is short, then either you don't do anything or you give them antifibrotic and avoid at all costs the immunosuppressive therapy. And if you look at the mutations for familiar cases of pulmonary fibrosis, you will find that in around 30 to 40% of the cases, they have mutations related to the telomere genes. So these subjects have short telomeres. And it's only 1 or 2% they have mutations related to the surfactant. So we know that in familial pulmonary fibrosis that they are enriched for rare variants. So in genomes, you have a rare variant where the allele frequency is less than 1%, but they have a big effect on the disease. Whereas if you have a common variant where it's more frequent in the general population, they have minimal effect on the disease. And again, not just in the familial pulmonary fibrosis, even when you take cases with IPF, you still encounter mutations related to the telomere genes. And if you take all comers with IPF, at least you will get 10% with rare variants affecting the telomere maintenance genes. But there are also common variants where they are commonly encountered in the general population. And if you take each variant on its own, then the effect is very minimal. But then if you put it as a polygenic risk score, so you put all the variants together and then you have the contribution of each variant, then at least you will have around 10 to 30% contribution to the disease risk. And the most common variant is the MUC5B, which was reported in the New England Journal of Medicine in 2011. And those with the polymorphism, which means that there's like a single nucleotide difference in the promoter region of the MUC5B. By the way, MUC5B is encoding protein mucin 5B. So if you have that specific polymorphism, if you get one allele, then you are something like 6 or 9-fold increased risk of IPF. And if you have the two alleles, then at least there are 20-fold increased risk of IPF. And this has led to the genome-wide association study, where basically it's a case control study. So you have all the patients with IPF, and you look at the variant, and you compare it to the control, the healthy subjects, and then you identify some susceptibility loci in the genome. So there are at least around 23 susceptibility loci identified in the human genome that is increasing the risk of IPF. And why is that important? Because in the future, you might find yourself using a calculator to calculate the polygenic risk score, because IPF is a complex disease like diabetes. Nowadays, there's even scores for calculating what's your risk of developing diabetes or ischemic heart disease. Then in the future, we might have in our hands such a score. And does one size fits all when it comes to treatment? So we all aware that in the Panther IPF study, there was a third arm, which is the N-acetylcysteine alone compared to the placebo. And when they analyzed the study, actually the N-acetylcysteine on its own compared to placebo did not change the progression of the disease. But when they looked at those subjects where they had the DNA samples collected on them, and they sequenced a specific gene called the TULIP gene, which is, has a role in terms of the innate immune system. So actually you find that there is a group of patients where they have the CC, which is like the nucleotide in that gene, then N-acetylcysteine was harmful compared to the placebo. If you had the CT genotype, then there was no difference. But if you had the TT genotype of the TULIP gene, then actually N-acetylcysteine was helpful in reducing the progression of the disease. And this has led to the design of a prospective study called Precision's clinical trial, where they will give N-acetylcysteine only to those subjects with the specific genotype, the TT genotype. And as far as I'm aware, I haven't seen the result of that. But they'll just show you the power of the genome in deciding not just the prognosis, but also in which treatment you administer to patients. And also it has been shown even with nintedanib. So this is taken from a registry in the Czech Republic. And when they looked at the genotype of a specific gene called the DSP gene, they found that those subjects taking nintedanib, and they had the G allele genotype, they had much better outcomes compared if they had the TT genotype. So it's not just one size fits approach. It's not one treatment to give into all fibrosis patients, but also it depends on the individual code of patients. And not just in terms of the treatment, but also the genomes playing a role nowadays and in the future, certainly, in helping in achieving diagnosis. So we know that we achieve the diagnosis of IPF, the usual interstitial pneumonitis, by integration of radiology, clinical, and also sometimes biopsy. But it has been shown and demonstrated that if you had looked at the gene expression at the tissue level, then that will improve the diagnostic accuracy in differentiating UIP from other forms of lung fibrosis. And you can see here how this will be integrated maybe in the future is to add some gene expression on the tissue level to help you in achieving the diagnosis. So this is a summary of the journey of the human genome. Started in 1990, the announcement of the Human Genome Project, and the first draft of that was published in 2001. And then in 2011, the FDA approved the next generation sequencing, which has enabled us to sequence the human genome rapidly and accurately. And in last year, there was the publication of the polygenic risk score for IPF. And this all started in 1950 with the first report of the familial pulmonary fibrosis in twin sisters, demonstrating the genetic contribution to the disease. If you wanted a summary slide of the presentation, it will be this one, because you can see here the power of the genetics in helping us in practicing precision medicine in terms of the screening, in terms of knowing the prognosis, achieving the diagnosis, in terms of which treatment to choose. Would you give nintedanib to the subject, or would you give pirfenidone or another medication? Would you give N-acetylcysteine to the subject or no? That all be decided by the individual genetic basis. If you are asking which patient that I subject them to genetic testing, at least the recommendation in terms of the publications of the European Respiratory Society is that you should be requesting whole exome sequencing on cases with familial forms of IPF. Familial means at least there are two members of the family where they have IPF, and also if they have an early onset of the disease less than age of 50 or 60, for example, and if you have manifestations of the telomere disease, which means that the liver cirrhosis and bone marrow failure, etc., then it's advised that these subjects should have the genetic testing. And I asked ChatGPT to draw me a picture to see how the practice of IPF will be in the future, incorporating genetics. And you can see that I think the way that we practice medicine nowadays, by giving the same treatment for everyone, every case of pneumonia receive the same antibiotic, every case of IPF receive pirfenidone or nintedanib, that I think is definitely changing. So that will help us in assessing the individual risk of the disease, assessing the risk of progression, develop preventive measures, early diagnosis, informing the treatment decisions, and also to develop new treatments. And thank you very much.